Hi everyone, welcome to our special Environmental Statistics Week. And this is organized by the Integrated Health Science Core of the Michigan Center on Life Stage Environmental Exposure and Disease. So starting from today, we will have 5 days in a row of the invited seminars. All these seminars will be dedicated to the current COVID-19 pandemic research, also the impact of COVID-19 on our general environmental health. So today we are very honored to have our first invited speaker, Dr. Yanxun Xu. Yanxun is an assistant professor of applied mathematics and statistics at Johns Hopkins University. Her research interests lie in a broad range of statistical problems, such as high-dimensional data analysis, non-parametric problems, uncertainty quantification, large-scale heterogeneous data problems, and also the electronic health record analysis. So Professor Xu just received a Faculty Development Award last year at Hopkins. She also won the Mitchell Prize in 2016 and David Breyer Young Research Award in 2015. So both of these awards are very prestigious awards in our field. Lately, after the pandemic hit us, she and her research group have already done a lot of work on COVID-related research topics. So today, she will talk about their findings on COVID-19 and racial ethnic disparities. So without further ado, Professor Xu, the floor is yours.
Okay, thank you, Lu, for your nice introduction, and thanks for inviting me to share my research on COVID-19. It's my great pleasure. Assessing treatment effects in hospitalized patients from underrepresented minority groups. So this is a joint work mainly with my PhD student, Quimbo Wang, and Dr. Brian Garibaldi, and also other clinicians and statisticians who are involved in Hopkins' analytical team on COVID-19 research. I would also acknowledge the fundings from Hopkins in Health, that's Johns Hopkins Individualized Health Initiative, and the Office of the Assistant Secretary for Preparedness and Response. So according to Hopkins COVID-19 dashboard, the pandemic caused by coronavirus continues to progress with over 40 million global cases and over 1 million deaths as of this morning. So those numbers grew more rapidly recently. So while the world awaits an effective vaccine, a number of different treatments have been started for treating COVID-19 patients. So those treatments have therapeutic targets against the COVID-19 at different stages of the infection. So for example, back in March and April, there were a lot of discussions on hydroxychloroquine. So it targets the viral attachment and endosomal acidification. And also we have drugs targeting, let's say maybe viral replication such as remdesivir, or the host cytokine response like tocilizumab and steroids. So globally, there are currently many clinical trials underway to evaluate the efficacy of a number of all those repurposed drugs used individually or used in combination against COVID-19. So in this work, we focus on the remdesivir. And here is a brief timeline for remdesivir development. And remdesivir was originally developed for Ebola, targeting a broad array of RNA viruses. So it has received considerable attention since its first effective case report on January 1st. And then China first started a trial at the beginning of February. The US started the trial on remdesivir on March 6th. After then, the April 29th was a big day for remdesivir. So Gilead first announced a similar effectiveness between 5-day treatment of remdesivir and 10-day treatment. But the China showed the drug didn't benefit severe patients. And then Dr. Fauci said remdesivir can shorten the recovery time. So despite the negative results from China's trial, FDA issued an EUA, that's Emergency Use Authorization, on May 1st, and then broadened the scope to include all hospitalized adult and pediatric patients with confirmed COVID-19. And then just 2 weeks ago, on October 8th, the final phase 3 report of remdesivir was published on NEJM. Okay, so currently there are already sufficient data demonstrating that coexisting conditions in patients with COVID-19 influence clinical outcomes, and also that older ages and male sex are associated with greater risk of death. And also people noticed disproportionality on a higher rates of COVID-19 infection, hospitalization, and death in racial and ethnic minority groups. So as we strive to overcome the social and the structural causes of healthcare disparities, we must recognize the underrepresentation of minority groups in COVID-19 clinical trials. So for example, in the Gilead trial, there were only 11% are Black Americans and 0.7% are Latino and Native American patients. And similarly for ACTT1 trial for remdesivir. So that has a higher proportion, but still it's less represented. And here is a figure about the racial and ethnic proportion of the whole population and the positive cases and deaths across the ACTT1 trial US states. And since these minority groups shoulder a disproportionate burden of morbidity and mortality from COVID-19, understanding the effects of remdesivir in those populations is very important. So to answer this question, we collected data from patients having confirmed COVID-19 infection from the Johns Hopkins Crohn registry. And this registry is securely hosted on the precision medicine analytic platform. It's called PMAP. And PMAP is a research discovery platform supporting data exploitation and analysis developed by the Johns Hopkins Individualized Health Initiative. Sources including the following. So it includes electronic medical record data from EPIC at Hopkins, and also the physiologic device monitoring system, and research electronic data capture is called REDCap, and also the biospecimen repositories. So between March 4th and August 29th, across 5 hospitals in the Johns Hopkins medical system, including Johns Hopkins Hospital at Baltimore and Howard County General Hospital in Columbia, and Suburban Hospital in Bethesda, and the Bayview Hospital in Baltimore, also, and Sibley Hospital in Washington, D.C. And we have 2,485 hospitalized patients with COVID-19. We can see here there was a higher mortality rates in Suburban Hospital in Bethesda and also Sibley Hospital in Washington, D.C. So to find out the reason why it has much higher mortality rates in these 2 hospitals, we plot the patient's admission dates by hospital. And we can see the Suburban Hospital has the highest admission proportion in early days. And when people didn't really know, how to treat COVID-19 patients as good as now. So this kind of partially explains the high mortality rates in Suburban Hospital in Washington, D.C., and also in the Suburban Hospital in Bethesda. And we also study the socio-demographic and other factors that could be associated with the proportion of severe patients. So here we plot the several potential risk factors for the disease, for the severe disease, like age, BMI, and the kidney disease, lung disease, and heart disease, and also the artery disease, and also diabetes, drinking, hypertension, liver disease, and sex and smoking status. So for example, we can see the age and kidney, heart, and artery, those, the pre-existing conditions, they are mostly significantly associated with the severe disease. And for mortality rates, those factors basically are consistent with the severe rates, right? So we can see the age, also the kidney disease, lung disease, and heart disease, these are still significantly associated with their mortality rate. But there are 2 kind of not really surprising results, but I think it's worth paying attention to is about the age and the hypertension. So we can see the age differentiates the severe disease and non-severe disease, but it seems it more significantly affects the mortality rate compared to the severe rate, and similarly for hypertension. Okay. Also, we plot the number of hospitalized patients based on their zip codes. And we can see the Baltimore City has the largest number of hospitalized patients, followed by Montgomery County and also District of Columbia, and also the Howard County. So if we see the number of severe patients by location and also the mortality rates by the location, we can see there, if you see the number of the proportion of severe patients, there's actually no significant difference on severe rates across different counties in Maryland. However, if we look at the mortality rates, and the Montgomery and the Washington, D.C. has the highest mortality rates. So we also want to know why. So these two counties has the highest mortality rate. And then we again plot these potential risk factors for the severity and also the mortality. And we can see here for the Montgomery County and also the Washington, D.C., and there's a significantly larger proportion of older ages, and also for the hypertension. This also consistent with our previous finding, the suburban hospital and also the Sibley Hospital in Washington, D.C. has the highest mortality rate. So the reason, because you see the suburban hospital was in Bethesda, so that's within the Montgomery County. And the Sibley Hospital is in Washington, D.C. So we can see, I think the age and also the hypertension contribute most significantly for the mortality and also the severe rates in these two counties. So about race and ethnicity, and here is the proportion of different races and ethnicity groups in COVID-19 deaths by county. And we can see the Baltimore City and the Washington, D.C. and the Prince George counties consist of a larger proportion of minority groups deaths, so like Black and Hispanic patients. This is actually related to the racial ethnic compositions in these counties. So for example, in Baltimore City, the Black American consists of 62.7% of the population. And also the Hispanic and Latino patients consist of about 5.7%. And also here's two races or more, and non-Hispanic takes up about 2.5%. So this kind of partially consistent with, you see the percentage of deaths also consistent with their general population compositions. So now we describe our data, related to the counties, their geographic locations, and also some potential risk factors. And now let's see how to assess the treatment effects in those patients. Okay, so we have 2,485 patients admitted to Johns Hopkins system. So we have 5 hospitals from March 4th and August 29th. We excluded the patients that either died or discharged within 24 hours after the admission. So this left us about 2,317 patients. And then we also exclude the patients who were involved in clinical trials back in March and April. And we also exclude the 4 patients who were still on treatment at the time of analysis. So we had 342 patients who used remdesivir. And also we have about over 1,900 control patients. Among all those patients, we have over 80% of the patients from minority groups. So if we look at the demographic information of all remdesivir patients and all control patients, we can see we have 36.3% of the Black patients using remdesivir and 33.3% Hispanic patients using remdesivir, and plus the other races. So we have over 80% patients from minority groups. So that makes our study kind of important because, the clinical trials, most of their patients are white patients, but our patients, most of them are from the minority groups. And also, if we look at their O2 device, so this is closely related to the disease severity. So for example, if no supplemental oxygen, that we call the mild to moderate patients. And also for all those, if people need the help on their oxygen, for example, the nasal cannula or face mask or some ventilation or mechanical ventilation, and we call all those patients as severe patients. So if we compare the remdesivir patients and the control patients, we can see a larger proportion of severe patients from patients who use remdesivir compared to the control patients. So if we want to estimate the treatment effects among those patients, then the disease severity of the O2 device is an important confounder when we study the treatment effects. So in statistics, estimate the treatment effect from electronic medical record data, so those are observational data, is an important causal inference research topic. And one of the methods is matching methods, so which aims to create a matching population so that this matched population can mimic the data from randomized clinical trials. And for this kind of methods, the procedure is we calculate the propensity score, and then, so this basically is the probability of receiving the treatment. And then a treatment and a control patient, they will match together if they have the same or very similar propensity score. So in our study, to study the remdesivir treatment effect, there's another complexity related to the different remdesivir received times. So if we just calculate their propensity score based on their patient's baseline information, it didn't account for the situation of different remdesivir received times. So for example, here, if we treat their hospitalization admission time at time 0, some patients, they receive remdesivir shortly after their admission times, maybe it's first day or second day. But some patients, they may receive remdesivir several days after the admission time, so even like 10 days after their admission time. So we cannot just only use their baseline information to calculate the propensity score. So how to adjust for the patient's baseline information as well as their disease progression before the remdesivir received times is a challenge here. So to analyze such data, we used a time-dependent propensity score matching method to balance the treatment on the control arms. So the way we solve this problem is we use the time until the received remdesivir as an outcome. And then we fit a Cox regression model using the time until received remdesivir as an outcome. So here we model the hazard for patients of receiving remdesivir at time t, and then we use the baseline covariates, so such as patient age, race, or BMI, and also the longitudinal, a lot of longitudinal covariates relate to the disease progression in this Cox regression model. And then we use the hazard component as a hazard for matching. So because here, the hazard component in this Cox regression model kind of is the probability of receiving remdesivir, right? So here, the matching covariates in our Cox regression model of modeling the timing of the receipt of remdesivir includes the following. So the time invariant covariates included age, sex, race, and the CCI, and also DNR, DNI, and the BMI. And the time-dependent covariates, they are mostly the risk factors for the disease severity and mortality. So for example, like the SaO2 over FiO2 ratio, and blood pressure, and pulse temperature, and respiratory rate. And also here, I want to point to the O2 device we just mentioned. So because the O2 device is closely related to the definition of the disease severity. So after we plug in those time invariant covariates and the time dependent covariates to understand the disease progression before the receipt of remdesivir, and then we calculate for each patient, we calculate that their hazard at time t, and also we calculate the hazard at time t for the control patients. We calculate that this hazard, and then we treat those hazards as a propensity score. Then we do the one-to-one graded matching without replacement. So that's how we kind of construct this matched population. We mimic the data from the randomized clinical trial. And here is our matching summary. So among the 342 patients, the 303 patients were matched successfully. And we can see their demographics. So here is the sex and the race and age, BMI, DR, DNI, and also the O2 device. This is closely related to their disease severity. And also we list the vital signs and laboratory results, and also past diagnosis, and also the co-used medications. So we can see even some of variables, we didn't really take them into account into the matching model, but mostly all those demographic and the vital signs all together, they were matched very well. Okay, so after checking the matching summary, and then we used the Cox regression model to take into account the propensity score and also a lot of other longitudinal covariates to study the effectiveness of remdesivir. And here are the results. Okay, so our primary outcome is the time to clinical improvement. So time to clinical improvement is defined as a WHO score decreased by at least 2 points or left discharge without worsening of WHO score. So WHO score, so if you're not familiar with WHO score, is when the WHO score is 3 or 4, that means it's mild or moderate. So that means those patients doesn't need the oxygen support. And if they need the oxygen support, then it's either 5 or 6 or 7. The WHO score being 8 means death. So we define the time to clinical improvement as decreasing by at least 2 points or left discharge without worsening of WHO score. So when we study the time to clinical improvement, the outcome first is rate censored at 28 days or max follow-up day, whichever comes first, because there are some patients, they stay in hospital for more than 1 month. So we censored all the data at 28 days. And the patients who died, so there's a competing risk problem, right? So the patients who died before the day 28 was censored at day 28. And so here is the results. And we found out remdesivir had benefits in time to clinical improvement. The hazard ratio here is 1.55, and 95% confidence interval is from 1.28 to 1.87. So here we also plot their cumulative improvement rate. Our secondary outcome is time to death. So for time to death, the outcome also is rate censored at 28 days or max follow-up day, whichever comes first. And for the discharged patients, so for time to death study, the discharged patients are competing risk patients. So for all those patients, if they were discharged before day 28, and then they were also censored at day 28. And here is our result. So we found out the result is not statistically significant. The adjusted hazard ratio here is 0.8. So remdesivir is helpful for the time to death, but it's not significant. And the confidence interval is from 0.46 to 1.41. And also here we plot the Kaplan-Meier plots of the survival probabilities. So this result actually is consistent with their ACTT1 trial. So that's their Gilead just reported their phase 3 trial on October 8th. So they found out the remdesivir has significant benefit on the time to clinical improvement, but they didn't have the significant benefit on the time to death. So this kind of complements the clinical trial results, right? So we have similar results as a clinical trial, but we have relatively different target population. And also we did further subgroup analysis stratified by severity. So remember, for the WHO score as 3 or 4, meaning the patients didn't need the oxygen support. And we further studied their time to clinical improvement and also the time to death. So we found out for the mild moderate patients, the remdesivir also benefits those patients. So they significantly shorten the time to clinical improvement. So here the adjusted hazard ratio is 1.54. And also we have the similar finding on time to death as the overall population. So the time to death is still helpful, but it doesn't help significantly. And then for the severe patients, so meaning that their WHO score 5 or 6 or 7, so those patients needed the oxygen support. And we found out the time to clinical improvement now, it became non-significant. So remember, for the overall population, remdesivir actually benefited the time to clinical improvement, right? So, and for the mild to moderate, they still benefit those patients, but now this significant benefit disappears if we focus on the severe patients, and time to death is still not significant. So this information tells us it's better to treat early. So because, the remdesivir, they can help the mild and moderate patients, but for severe patients, this significant benefit disappears. So regarding the remdesivir, a lot of people also concerned about their adverse events. So for example, people concerned about the side effects of remdesivir on people's liver function and the kidney function, especially the liver function. So to study the side effects of remdesivir, we also summarize their adverse events related to the liver function. So here represented by ALT or AST larger than 200, or the bilirubin larger than 2 milligrams, and also their index related to the kidney function, the EGFR. And now we can see the matched remdesivir and also the matched control patients, actually, they were similar, right? So even the matched control have a small, very tiny increase of their adverse events compared to the matched remdesivir. So this tells us those side effects could come from disease itself, not necessarily the drug. So it kind of ensures the relative safety of remdesivir. It doesn't have significantly higher rates of adverse effects compared to the matched control patients. So the NEJM paper also reported such similar rates of adverse events in the remdesivir patients and their control patients. So besides the remdesivir itself, so we also want to study the drug combinations. So especially the remdesivir plus the one type of steroid is dexamethasone. So because the dexamethasone as steroid, it targets people's inflammation, also draws a lot of attention, from April. And if we compare the remdesivir plus dexamethasone patients versus remdesivir alone, so we have 157 patients who use the remdesivir and dexamethasone, and 185 patients who only use remdesivir alone. And if we compare their demographic and also the race ethnicity, we can see the remdesivir alone, we have a relatively higher proportion of Hispanic patients, but similar Black patients. And also, if we compare the time to death and time to discharge, the remdesivir and the dexamethasone patients have a slightly lower, shorter time to death and also the short time to discharge, but those are not statistically significant. And also for the baseline disease status for the remdesivir plus dexamethasone patients, they are actually similar to the remdesivir alone patients. And also if we look at the death and also the discharge, so we can see the lower rate of death for the remdesivir plus dexamethasone patients compared to the remdesivir alone. So this kind of warrants us to further study the data to compare these two groups of patients. So here we show the time cost for the co-administration of remdesivir and the dex for the 157 patients. We can see most of the patients, they either received remdesivir and the dexamethasone at the same time period. So here we have yellow color for the, sorry for the low resolution of this figure. So we have yellow color represent the dexamethasone and blue color for the remdesivir. So we can see they either receive a similar time period or people receive remdesivir first and then dexamethasone. As a majority of the patients received the 6 milligrams of dexamethasone daily. Okay, so we further used our model to compare the remdesivir plus dex patients with remdesivir alone. So remember, when we study the remdesivir patients, we use the propensity score matching method. But here, since remdesivir patients, we only have a small number of patients, about 300, so it's not feasible to do the matching method. So here we used the marginal structure Cox regression model to study the drug effect of the remdesivir plus dexamethasone patients compared to remdesivir patients alone. So we use the marginal structure Cox model via the inverse probability of treatment weighting methods. And then here is a result. We found out regarding time to clinical improvement, the hazard ratio, it doesn't change much. So here is 0.87. And the 95% confidence interval is 0.53 to 1.44. So we didn't see significant difference between the remdesivir plus dex versus remdesivir patients. But if you look at the time to death patients, so for the time to death analysis, just hazard ratio is 0.14, and 95% confidence interval is from 0.02 to 1.03. So we can see the upper bound here. So the upper bound is crossed to one, it's trend towards mortality benefit of the dexamethasone, right? So now we have this marginal benefit of the remdesivir plus dexamethasone versus remdesivir. It gives us a trend towards the improved mortality. So if we still remember the results from our study and also the ACTT1 trial on remdesivir, we know the remdesivir can significantly help the time to clinical improvement for mild and moderate patients. So it raises a question. So about the preliminary results show that the dexamethasone as a steroid, it only benefits the patients receiving supplemental oxygen or additional support. So that means the dexamethasone only benefits the patients who are severe. So those are needed the oxygen support help. But for the patients on room air, so that means for all those patients who are either mild or moderate, they actually did not benefit from the dexamethasone. So it raises one critical question. So since remdesivir benefits the mild, moderate patients, but dexamethasone only benefits the severe patients. So when we apply the dexamethasone and remdesivir together, so for those co-administration of those two drugs, what is the appropriate target population? And also for the co-administration. So that's a very challenging statistical question. So currently we're developing a dynamic treatment regime method to answer this question, but due to time limit, we will not show any technical details. So we're showing, what's the optimal sequence of if you want to apply the remdesivir and dexamethasone, and also what's the optimal timing for co-administration of those two drugs. Okay, so to summarize the talk, we know currently the optimal implementation of therapeutics to reduce COVID-19 morbidity and mortality is a global priority. So we need to acknowledge the racial disproportionality in COVID-19 clinical trials. So our study learned the treatment effect, especially remdesivir, so we can apply the similar methodology to other drugs. For example, like we have another work on tocilizumab. So we learned the treatment effect of remdesivir from this retrospective electronic medical record data with over 80% of patients from minority groups. So since the underrepresented minority have shouldered a disproportionate burden during the COVID-19, but they haven't been widely represented in clinical trials, our study complements the clinical trial results. So showing that remdesivir also benefits these populations. And in this study, we also examined the co-administration of remdesivir with other drugs, especially the dexamethasone. And we found out, compared to the control patients, remdesivir and the dexamethasone had the significant benefits on time to clinical improvement. But however, if you compare remdesivir and dexamethasone versus remdesivir alone, then you didn't find significant benefit on time to clinical improvement, but we have a marginal benefit on the time to death. So the future work is, about how to optimally implement the treatments and also their combinations. So before the end of the talk, I want to mention several challenges on how to optimally implement the treatments and their combinations. So in our data set, we have over 200,000 treatment records. And for those records, within those records, we have 473 different treatments or drugs in 300 different subclasses. So for example, like, these subclasses include ACE inhibitors or statins or antibiotics or steroids. And because most of patients, they use the drug combinations. So in our data set, we have over 6,000 different drug combinations. So it imposes very serious statistical challenges in assessing this drug combination effects, right? Because we only have a little over 2,000 patients, but now you have over 6,000 different drug combinations. And also, if you study the individual patients, the number of treatments varies a lot among different patients. So the mean number of treatments used per patient is 145. So we have about 145 treatment records for each patient. And the maximum of treatment records for one patient is 1,505. So this is really a large number, right? And the minimum treatment record for patient is 1. And not only you have a huge number of drug combinations, we have these 6,470 drug combinations, and also over 95.6% of patients, they have drug switches during the hospitalization. So that's kind of, the dynamic treatment regime questions, right? So patients receive a sequence of treatments, and you want to estimate what's the optimal sequence of treatments to treat those patients. Another challenge is how to incorporate the demographic and social environmental variables in clinical decision making. So we showed the number of cases in different counties. So we know the different counties, they have different demographic and environmental variables, right? So for example, different counties, they might have different racial proportions, and they might have different diets and also other more environmental habits. So also for this complex decision-making problem, with this huge number of drug combinations and also the frequent demographic, social, environmental variables in the clinical decision-making is also a very challenging statistical problem. Okay, so thank you. So that's all that's all for my talk. It's right at time. It's about 40 minutes. Yeah.
That is a great talk, actually. So when you talk about dynamic treatment regimes, are you talking about to develop some clinical trials with re-randomization among the patients, or you're only using the observational data to evaluate those dynamic treatment regimes?
So the current work is just analyze the electronic medical record data. But our Hopkins group, so our analytical team actually were currently developing some tools for the risk prediction in hospitalized patients. But those risk prediction tools, we didn't really consider their treatments. So after we analyze this electronic medical record data, and then our future plan is to also incorporate those methodologies to the risk prediction and take into account if you use those treatments, what's the risk for the disease severity and also mortality. And then that might help. So I'm not sure now, but I think that maybe some prospective clinical trials we can plan based on all those tools.
Mm-hmm. Yeah, I think like you mentioned, the environmental risk factors and also their habits, all of those information are very hard existing there in the electronic record right now. But probably when you design the trial, you can collect those information and see how the impact of those environmental risk factors are. Yeah. That is a great talk. So I'm going to turn to the audience and see whether they have other questions. Audience? I guess
my talk is crystal
clear. Exactly. That's what I want to say. Talk is very clear. So Yanxun, you mentioned earlier about the figure all the maps. So I wonder whether you incorporate any special analysis in your data analysis, and also the disease mapping, whether that will help us in this scenario. I guess probably to account for the spatial temporal correlation is important, but in the meanwhile, because this is infectious disease, so then they have many other, like the distance, the social distancing, and all those measures, wearing masks. So those matters as well. So I guess most of those still belong to the environmental habits, in the community or in the neighborhood.
Yeah. So it's very good thought. So at the beginning, the results actually kind of was a little bit surprising, because like Montgomery County is a relatively rich county in Maryland.
Yeah.
So we didn't expect that such high mortality rates. But then we analyzed the data. So like I said, so actually, because at the beginning, our treatment is not, our knowledge about the treatment is not as good as now. So those hospitals have a larger proportion of patients from the earlier days. And also, I think the most importantly is at the very beginning, there are a lot of patients from nursing home in Montgomery County, and also the Washington, D.C. So, previously we tried to find out, is there any environmental difference, and also there maybe, the racial difference or ethnic difference, that contribute to the high mortality rates. Mm-hmm. But after the careful study, and we found out actually the age is contribute significantly to the different mortality rates among different regions in Maryland. But I think now, after May, so when we know better about their treatments, and also, the population kind of also shifted from relatively older patients to younger patients after May and June. And I think, all your questions maybe can answer them more. I think it's kind of like, we adjust for, or we exclude this, the factors about the age, and also their, the age, and also their, the locations. I think we might be able to know about the environmental effects on the mortality and also the severity, disease severity rates.
Mm-hmm. Yeah, I think the same.
Sorry, my screen just showed the internet was not that stable, so I hope you can hear me clearly.
Yeah, we did have some glitches, but overall it's okay. So what I want to say is, in Michigan, we also have similar studies. And I think the major thing at this time is, because this is very kind of urgent issue, and it's kind of emergently collect the data from the electronic health record, or here we call them chart. So from the chart data. So they didn't collect enough about their environmental risk factors. But we did think about those. Then when we look at the data, we will try to search, do we have this? For example, the social relationship, or the poverty level of the neighborhood, and the behavior, like the habits. And we want to see, do they have this in the neighborhood? But then very unlikely that we will have those information. So I think later on people start to collect this data, but may take some time. Yeah. I do have another.
Right. Also, I think,
yeah, go ahead.
Sorry. Yeah, I mean, so I think it's also a very good direction, of the study, because so I think it's also related to how to, integrate data from different sources. So like you said, from the hospital perspective, it's very hard to collect the data about the environmental or, some other like social demographic factors, right? So I think, Hopkins, also in our project, like we try to integrate the data from the medical system And also, for example, the epidemiology, Professor Lauren Gardner's, the Hopkins, COVID-19 dashboard data. And also we actually have a lot of environmental databases, like, about either the pollution, air pollution, and also, the poverty and all those things. We have all those data sources, even from the county level. It's not really the individual level. But I think for those environmental factors, the county level data might be already good enough. Right. So I think the key point is how you can integrate the data from the medical system and also from all other different data sources.
Mm-hmm. Yeah, definitely. So Alan has a question. So maybe Alan, you can unmute yourself.
Yeah. Can you hear me?
Yeah.
Yeah.
Thank you so much, Professor. This is a really great and interesting talk. I had a question about some of the first Kaplan-Meier curves that you presented for the matched analysis.
Yeah. So this one?
Sorry, there's a little bit of a lag. Okay, yeah, I think the next one.
The death or the
time to death,
one?
Yeah.
Okay.
So in this figure, so since you included when constructing the propensity scores all time variant and time invariant confounders, is that correct?
Mm-hmm.
So
yeah.
The Kaplan-Meier curves that are generated in the matched analysis are essentially akin to what would be in a randomized trial, right? Or trying to get as close as possible to that. Is that correct?
Yes.
Mm-hmm.
Okay. So is it fair to say that you can sort of interpret some of these Kaplan-Meier curves in this beyond what you might interpret to be the average hazard ratio? So it seems like it might make a difference later on here. Am I understanding that correctly? Or is that a fair assumption to say that remdesivir actually might make a difference to patients that survive later on?
Sorry, I think there is some lag in the internet connection.
I can type the question if it's better.
Yeah, Alan, I think, typically for this, if you use propensity score matching and then the Kaplan-Meier, you can put on the inverse weighting of the probability to be in the code, and then that can account for that. So after the weighted version, basically is try to mimic a randomized trial. And then if that, the Kaplan-Meier is shown to is already accounting for the inverse probability weighting, and that is a fair statement you just said, I think. Yeah.
Right. So it might provide information beyond just looking at one single hazard ratio.
Exactly. Yeah, and also for the log rank test, which is corresponding to Kaplan-Meier, right? So the test that also can allow you to put on some weight there. So typically, inverse probability of the propensity score, that weighting can handle this type of thing when you does not have a randomized trial. Yeah.
Right.
That's a very good question.
It seems like that might have been the case for a couple of the outcomes where the effect of remdesivir did change depending on the time, I guess, at which it was administered, or the time until.
Yeah, I think, this basically happens, in this case, is time variant weights, but some other case, the probability of assigning the treatment is one-time thing, so you just have time invariant weight. But for this one, you have time variant weight, and then the package right now, they can handle that time variant weights. Yeah.
Sorry, I think my internet has certain problems, so I log in using my phone and also keep the Zoom on the laptop. So Lu, you answer the question for me, or?
Well, I answered the question not on you for your for your specific data analysis, but I believe that you did the same thing. So basically here you have observational data. So then the treatment is not randomized. So you need to account for the potential confounding for the treatment assignment. So basically either you control for the propensity score or you use inverse probability weighting to wait for the inverse of the propensity scores. And that Kaplan-Meier, I believe you have already account for that, right?
So the Kaplan-Meier actually it's just the empirical Kaplan-Meier plots for these two groups. But when we do the analysis, we actually adjust for all those things. So the adjusted hazard ratio, actually we have already adjusted all baseline covariates and also the longitudinal information. But the Kaplan-Meier is just just regular Kaplan-Meier.
Original data. So
okay, that's what I was wondering.
Yeah. So Yanxun, do you know that in the R package or SAS, you actually can use the weight in the option to incorporate the inverse probability weighting. So that will give you a modified version or inverse probability weighted version of the Kaplan-Meier. And then that also the log rank test as well. That will be much better to present, right?
Yeah, that's good to know. Thank you.
Sure. Hey, so Yanxun, I do have another question. Unless other people have a question, I want to ask you. So we also have a very similar issue as you, but you do not have a very severe issue of this. So when you talk about the patients, you have over 2,000 patients, and then you consider the treatment. Then I noticed at the beginning you exclude those who are in trial. So you have only 12 people who are in trial. However, Michigan data, when we have a limited number of patients at the University of Michigan, then we only have about 300 or 400 some people by the time like July. And then in the meanwhile, because that is at the beginning of the pandemic, when the patients are in the hospital, we actually enroll, I mean, not us, but then the physician, maybe some other physician who are running the trial. So they enrolled most of them into the different trial of the different new drug development. So then, because this is a clinical trial and most of them are blinded, so besides the treatment that we are interested in and we do the analysis, even using propensity score matching, however, there is underlying, unmeasured confounder because they may be treated by something else, which is blinded. So we didn't even know what the information are at that time. It's double blinded. Yeah. Do you have any other, like thoughts how to deal with this?
So I think so. Your concern actually the reason why we excluded the patients in clinical trials, because we don't know what really happened, and also the trial, many trials are double blinded, so that's why when we do all those analyses, we just excluded all those patients.
I see. I think excluding will be the best way.
Right. And I think luckily for us, we have a large enough population to study the problem, and we only have a small number of the clinical trial patients, so we just exclude them.
Yeah. I guess another option probably is at the end of the the other trials when they unblind the treatment options, and we can control for that later on.
Right.
Yeah. It's very complicated.
Yeah, that's true.
Well, do we have other questions from the audience? All right, so maybe we can wrap up this. And that is a very, very great talk, very interesting. And thank you for all the contributions to these research topics. I think it's saving a lot of people's life with your collaborator and physicians together. So wonderful job.
Yeah, thank you.
Thank you very much, Dr. Zhu. That was fascinating. Thank you.
Thank you. Yeah.
Thanks everybody for coming.